Okay, thank you very much. And also thank you very much to the organizers. It's great to be here. I'm enjoying the meeting quite a lot so far. I hope you do as well, and I hope my talk won't change that. Anyway, it's a great pleasure to be back here and to tell you a little bit about transcription and translation. As you just heard, this will be a series of 3 talks, 1.5 hours each. So the idea is that this is a bit tutorial and that you learn something. If you feel you don't learn anything, tell me. If you have the impression you don't understand anything that I'm talking about, please also stop me and ask. Yeah, I think that's what I wanted to say. Before I start, I wanted to show you where I come from. This is Göttingen. Göttingen is right in the center of Europe, really pretty much in the center, and it's, sorry? No, you can't. Yeah. I think we should discuss this over a beer later rather than now. Anyway, so it's a very old university. Well, not very old. It's not a medieval university. It's a university that was founded later, but it's also somewhat old. You see we have some old buildings, and I'm showing you this because you heard about the most famous professor of Göttingen earlier today. That was Gauss. It turns out that Gauss was a professor of astronomy in Göttingen, and this is the old observatory that was built for him. I think today this is a meeting place, conference place. Here you see Gauss. He was depicted on the old 10-mark bill. And you also see that they didn't put anything about curvature on this bill, but rather they put a Gaussian. And if you look very closely, you can even see the equation here. Maybe you can't see this on PowerPoint, but if you had the actual bill, then you could read the equation. So if you didn't remember the equation, you could just look it up on your money. And behind the equation, here, this thing you see here is the city of Göttingen. Okay, so that's what it looks like. It looks like a nice medieval city. And, well, there is a very nice university there and some very nice science. Okay, this is my research group. I just told you that Göttingen is a somewhat old university, so every old university has some strange traditions, and this is where we are celebrating one of these things. So you see here 2 PhD students that have recently graduated, and one of the things that PhD students that graduate have to do by tradition is that they go to this fountain, which is right in the city center, and the fountain has a statue, which I think you can see here. Here you see the statue of the goose girl, and every new PhD climbs up onto this fountain and brings flowers to the goose girl and kisses the goose girl. So this everyone has to do. I have no idea why, but that's what the PhD students do when they just graduated. Okay, but the important thing here is I'm showing you the group. Well, now you see only half the group, because you need a group to do a lot of work, and actually a lot of the credit for everything that we do goes to all these people that you see, and, well, some of which you don't see. Anyway, here's a very quick overview of what we work on. We work on a variety of different topics, which I grouped into these 3 groups. One is motility and active matter. Mostly in the last years, we have worked on magnetotactic motion in bacteria. And I think there was the ICTS online meeting last year or 2 years ago? 2 years ago, and if I remember correctly, I gave a talk about this in that meeting. And maybe the video is still online. You could even look this up if you want. Then we work on various types of stochastic dynamics in cells, some of it related to translation and transcription, which I will talk about, and on gene expression, cell growth, and population dynamics, which is also related to these stochastic dynamics problems, at least in some aspects. Okay, so here's what I'm planning to talk about in these 3 lectures. The beginning is pretty much fixed. This is what I talk about today. The end is still a little bit flexible because I might change it after this lecture or after tomorrow's lecture. So the plan is today I'll start with a general introduction into gene expression and the central dogma. Some of these things you have already heard in previous talks, some maybe not. And then most of today's lecture I will talk about speed and accuracy of processes like transcription and translation. And then the plan is that tomorrow I'll talk a bit more about the theoretical background of these problems, a more general theoretical framework for discussing this, stochastic dynamics on networks, and then about coarse graining and show you some work that we have done recently, how to work with this stochastic dynamics on networks. And then the third lecture, currently my plan is that I will take a somewhat different perspective and talk about the economic decisions that a cell has to make with respect to these processes. And there is a point 6 here. Maybe I will talk about something else if I feel like it somewhere down the line. Okay, and again, please interrupt me anytime if there is a question. Okay, I can talk by myself for a long time, that's not a problem, but it might make more sense if occasionally there is something that comes from you. Okay, let's go. So the first topic is gene expression and the central dogma. And if you have paid attention in the last talk, then you have seen some of this already. So this is my graph of how genetic information is read out. We have a DNA. This is typically one or a few molecules in a cell, very big ones, and there are small pieces on it that are genes. And these basically are the blueprints for individual proteins. And to make these proteins, the cell has to carry out 2 steps, 2 processes, transcription and translation. In the first step, the information from this DNA sequence is copied into a messenger RNA, and then in the second step, the messenger RNA is translated into protein. And the proteins basically do most of the work that is to be done in a cell. Okay, this simple rule that the information flows in one direction, starting from DNA to RNA to protein, this is called the central dogma of molecular biology. And I think in the next slide, no, this is not in the next slide. Well, I first should mention there are 2 molecular machines that do these 2 things, RNA polymerases and ribosomes, and you have already heard about those as well in the last talk, maybe even before. Anyway, what I wanted to briefly show you is this is Francis Crick's early draft where he coined this term of the central dogma. Here he writes the central dogma: once information has got into a protein, it can't get out again. So there is this one directional flow of information from DNA towards protein. What is somewhat funny about this is apparently Francis Crick at the time when he coined this term didn't really know what dogma means, because dogma in the Catholic Church is something that is true by authority of the Pope or of the Church. Okay, it's the most non-scientific thing that you can imagine. But somehow the name has remained. So this is still called the central dogma. I hope every time you hear this, now you sort of have a slight grin on your face and know, well, this name is a bit weird. Okay, this is the process in a little bit more detail. Sorry for this slide. This is, first of all the labels are in German, and second of all, this is some ugly drawing I made years ago, and I always think at some point I should make a nicer one, but I never do. But it shows you the process in some more detail. Okay, this is the DNA that you have seen before. There is a gene depicted as an arrow, but basically this just means it's a part of the sequence. Okay, there's this long string of A, T, C, and G, and there is one part of that that is the part that will be used to make a protein. And what happens is RNA polymerase will bind here at what's called the promoter, and then it moves along the gene, which you see here, and step by step, nucleotide by nucleotide, copies the information into the RNA, which is this one here, basically comes out in parallel in quotation marks. So you get a sequence that, a molecule that has copied the sequence information. And then in the second step, basically the same thing happens again if you don't look at the molecular details. You have another molecular machine, the ribosome, that comes on a start site, and Debashish can now tell me this is not correct because the ribosome is not really, these two parts are not together before it comes on here, they only come together when they are there. But anyway, let's ignore this for now. You have the ribosome come on here, move along the sequence, copy the sequence, and thereby generate a protein. The protein eventually has to go into some three-dimensional structure to do something. Okay, this is the process. So what we are going to talk about today is a bit more the details of this process, and in particular, this process of copying sequence information. Yeah, you have this machine moving along that sequence, copying the information that is there and making a copy of that information in a new polymer. And so one of the questions is how accurate is that? And another question is how fast is it? Okay, there's one more slide where I show another version of this central dogma. So you see here in the middle this one-dimensional flow of information. DNA goes to RNA, goes to proteins, but then you see lots of arrows that point backwards. Okay, and this is not copying of sequences, but what it shows you is, well, actually, to copy the sequence information from DNA to RNA, for example, you need an RNA polymerase. And this we have also heard in the talk before. The RNA polymerase itself is a protein. So there is something from back here that is needed all the way in the first step. So there is something that loops backwards here to actually carry out the process. And there are other things that loop backwards, and these are regulators. You have transcription factors that control transcription. You have other regulators that control translation. You may have RNA regulators that control transcription and translation. And of course, the ribosome itself, the machine for the second step, that is also produced by these two machines. The ribosome is a big complex that consists of both protein parts and RNA parts. So you have one arrow coming from here, and you have one coming from there, and well, needed in this. So if you look at the network this way, it doesn't look quite that one-dimensional anymore. That's just there, but you have this one-dimensional flow, but then you have added on top a lot of sort of loops backwards, feedbacks that are needed to make this work. Okay, just very briefly, why do you need regulation? Regulation is needed for a lot of things, and maybe the most obvious cases is that if the environment changes, cells adapt to the environment, and then, for example, they need different enzymes to process different types of nutrients. Whatever, I came to India, now I eat Indian food, maybe I need different enzymes, I'm not sure. I'm sure that this happens with bacteria. If you feed them different types of sugars, they will make different enzymes. Okay, another option is stress responses. If you put an organism into an environment that is very stressful, then there will be other genes that will be used that were not needed before, but are needed now to cope with that stress. Another option is developmental programs. So in bacteria, that would, for example, be when they make spores or when they make biofilms. Or sometimes it could be differentiation, which is certainly very important in our body because we have different cell types that have exactly the same genetic information but do completely different things. Think of a cell in your brain and a cell in your heart. They have completely different tasks. And for that, well, basically you have a differentiation. And this could come in different forms. It could come in the form of a division of labor, different cell types to do different things and work together effectively, which is what they do in our body. But it could also be bed hedging where different cells do different things because, well, basically they just try out different strategies to survive, which is, well, also something that makes sense, I guess. Okay, I will not talk much about this, but I thought I'd include this for background. Okay, let's get to the main part of what I wanted to talk about today. So this is about copying the genetic information from DNA to RNA and later from RNA to protein, and ask about speed and accuracy of these processes. Okay, so this is the general process. Again, I now included this loop for DNA because if you think about DNA replication, that's again a process of this type where a sequence is taken and copied. In that case, actually just copied from DNA into a new DNA. And all these processes, if you imagine them as just some type of chemistry, these are polymerization processes. You make a polymer, and they are copolymerizations because you make a polymer that has different types of monomers. And importantly, this copolymerization is not random, but it's directed by a template. So you have one polymer and you copy the sequence, which means you make a new polymer that has exactly the same information. And it happens in all these different cases, even though molecularly these are quite different. It happens by an enzyme that steps along the template, really moves along the template, which is also why we can think of them as motors. And then it inserts one monomer after the other into the new polymer. And in doing so, it has to always pick the monomer that is needed according to the template sequence. Okay, so it needs to recognize in the template sequence which monomer to use and then do the chemistry to insert it into the new polymer. And then you get this new polymer that has the same sequence information. And of course, that's not all. You need some additional processes to start this whole process, to get the polymerization started, also to stop it at the right point. And all of that also doesn't work by itself, but it needs some energy input. And the energy input typically comes from some high energy chemical bonds. So these are usually phosphate bonds in either the monomers themselves or in additional GTPs that are consumed on the way. Okay, let's now start with the very, very minimal description that we can make of this process. And again, I hope you remember this from yesterday. Yesterday you heard about the Michaelis-Menten kinetics in much more detail than what I'm going to tell you today. So Michaelis-Menten kinetics is basically the simplest type of kinetics that you have in biochemistry. Also, almost everything can be described as this, as Michaelis-Menten kinetics, unless there is some reason not to do it. Okay, so let's start with this. And what you have in Michaelis-Menten kinetics is first a reversible binding of a substrate, and then an irreversible transition that converts the substrate into product. And that's exactly what our machines do. I've shown this here for the ribosome. You have the ribosome, that is the enzyme. It takes, the protein the growing protein chain that it has, plus the new substrate that comes in, which is a charged tRNA, so a tRNA that carries the amino acid that is now needed for elongating the protein. This binds to the ribosome, and you can imagine this as reversible, and then this new amino acid is incorporated into the protein chain, and the ribosome is ready to do the next step. Okay, so you have these two steps here in this very simplest description, and it basically is textbook Michaelis-Menten example. Again, you can do the same thing for the RNA polymerase or the DNA polymerase, where you say, well, the polymerase is there, a new nucleotide comes in, and then it gets inserted into the growing RNA chain or DNA chain in the DNA polymerase, and afterwards the polymerase is ready again for the next step. Excuse me. Okay, if we look at this process, you have seen some derivation of this kinetics yesterday, but I want to briefly show you a very simple derivation of this kinetics, because basically what we'll do in the following is extend this to slightly more complicated cases. If we look only at the enzyme, then in this process here, the ribosome or the polymerase has essentially just two states. It's either occupied, something is bound, or it's not bound. Okay, that's what's happening. The tRNA comes in, and then it's occupied, and either it unbinds, then the enzyme is free again, or this gets incorporated into the chain, and the enzyme is also free again. So effectively, we just have a two-state system. We have the free enzyme, and we have the occupied enzyme. We have binding. So this is the reversible binding. And then in addition, we have this transition in which the new monomer is incorporated into the chain, and we also go back to the free enzyme. Of course, in this transition, we go to the free enzyme with a new polymer that's longer by 1 unit. Okay, so this process with the rate omega -1 and the process with the rate omega 2, they are different, but from the point of view of just looking at the state of the enzyme, they lead exactly to the same state. It's free again. Just the length of the protein, the growing protein chain is different. Okay, and so this two-state model you can easily solve at the rate equation level, and then you get this kinetics. They say that the rate of the overall polymerization is just, the chain is growing whenever this transition happens, and this happens whenever with the overall rate of omega 2 times the probability that the enzyme is occupied. Is this clear to everyone? I find that yes a little bit too weak. Can you either say yes more clearly, or I don't trust you right now. Okay, so the idea is we have two states, okay? 1 and 2. This is the same thing as up there. And each state has a probability, P1, P2. And what we're doing then is just we write down the balance of these rates. So what do we have here? P1 is this one here. So from P1 to P2, we have a omega 1. And backwards, we have omega -1 P2 plus omega 2 P2. And we have a second condition, namely P2 plus P1 must be 1. Okay, this is the amount of math that we're doing today. The only thing that will become more complicated in the following is that instead of 2 equations, we will have 3 or 4, maybe 5, I'm not quite sure. Okay, so these are our equations. They are nicely linear and easy to solve. And basically we're just interested in the flow through this arrow here, because that's how the chain grows. And the flow through this arrow is omega 2 times the probability of the occupied state. And if you solve these equations, then you get what I've written here. And this is the form that you hopefully remember from yesterday, where you have a Michaelis constant, and the Michaelis constant is just the collection of these parameters here, okay? Everything in purple. I have written this omega 1 as some rate kappa times the concentration, because this is binding, so it should depend on the concentration. And if I take the concentration out and collect everything else into a constant, I get this Michaelis-Menten equation for the flux of that relation, of that reaction. Okay, one small point. Oh, yeah, there's a drawing of this, how this looks like. For small concentrations, this is just linear in the concentration, and then it saturates. Nothing too exciting there. Okay, one point that I wanted to make is that this Michaelis constant, it's a ratio of the two rates that go towards the free state to the one rate that goes to the occupied state. Okay, and it's very similar to a binding constant, or more precisely to a dissociation constant. But if you take the dissociation constant for binding, then in an equilibrium situation, then you wouldn't have this. You would only have these two processes. Okay? And you can approximate this Michaelis constant as a dissociation constant in the limit where this rate is very small, where basically your substrate binds and unbinds many times before it gets incorporated. So the first step is basically in equilibrium before the second step happens. In that case, the Michaelis constant is equal to the dissociation constant. But in general, it's not. In general, these are two different things. This is an equilibrium property. This is not. Even though it looks pretty much the same. If you think of this as just mathematics, then this could be exactly the same equation that you get for, let's say, a Langmuir binding or some binding kinetics. Okay, but the nature of this constant is different in the two cases. Okay, now what we want to do is we want to look at a process where a sequence is copied. And again, I'm going with a somewhat minimal description. Okay, we don't really care about the sequence here. We just say, well, whenever something is copied, it can either be copied correctly or there can be a mistake. Which means in my kinetic scheme, I now have two such Michaelis-Menten schemes in parallel. One for the correct copying and one for the incorrect copying. So I have binding of the correct new monomer, I have binding of the incorrect one, and then I have incorporation in both cases. And now I have one state more, so I have now three states. I do the same math as here, just with three equations. They're still linear. You can still do this, and you get the probabilities of all these states. And the question now is how fast does this chain grow? Okay, and the chain now grows whenever something is incorporated. This could be a correct monomer, or it could be an incorrect one. Each time the chain gets longer. So the overall velocity of the reaction is now the sum of the fluxes through this arrow here and through this arrow. Okay, so the polymerization velocity now has two terms, VA, PA. So VA is this probability, this rate. PA is the probability of the correctly occupied state, plus VB, PB. VB is this rate, and PB is the probability of the incorrectly occupied state. And if I solve my three linear equations and put that in, I get this expression here, where the H now stands for basically concentration divided by Michaelis constant, either for the correct branch or for the incorrect branch. Okay, what have we now got? Well, we could, for example, now ask, if we have incorrect monomers instead of correct monomers, does the reaction get slower? Or does it get faster? If both are incorporated at the same rate, then having incorrect monomers in addition, of course, that will make the reaction faster. Did you want to say slower? No. Okay. How can you make it slower with the incorrect ones? When the incorrect one does not bind? Oh, you mean in the second step, in the incorporation? Okay, so you're saying if this step still happens, but this one does not. Yeah, then, I mean, if you just put VB to 0, then, well, this term is gone, but you still have this HB down here. Okay, so in that case, it will be slower. And typically, this VB will not be 0, but it will be smaller than VA. Okay, that's what you typically have in these enzymes. So you could expect that the polymerization gets slower if, in addition to the correct monomers, you also have incorrect monomers. And, well, if they're not incorporated at all, that's, of course, very good for the accuracy. Yeah, that's, in principle, that's what you want to have, because eventually you will have a product that is correct. But then it also gets slower. That's a relatively general principle that usually accuracy and speed don't go together too well. There's usually a trade-off between the two. Okay, let's go back here. So we can quantify this, how much the incorrect monomers slow the process down, by saying, well, we take this phi, which is the ratio of the speed divided by the speed if we don't have the incorrect monomers. And, well, if I approximate a little bit, I get a somewhat nicer equation here. And what I already said is typically VB is smaller than VA. Typically, incorporation of the wrong monomers is slower than incorporation of the correct ones. And that, in general, means that this phi is smaller than 1. Because this, well, this is smaller than 1 here, and this is the same above and below in the fraction, so there will be a slowing. So if this is small, the only way of making this phi close to 1 is if this HB over HA is also small. Okay, if this is very small, then you still have this down here, and this is what slows down the process. So this has also to be small so that the ratio as a whole remains close to 1. So what we need is that HB divided by HA is small, and if I spell this out, remember this H was just concentration divided by Michaelis constant. This means CB over KB must be small compared to CA over KA. And if you imagine a cell, then the concentrations of the correct and incorrect monomers, this will be different amino acids for the ribosomes or different nucleotides for the polymerase. They will have similar concentrations. Also, they exchange roles because they have a sequence. I mean, what is correct in one position will be incorrect in another position. So you cannot make the incorrect ones small in concentration. Okay, so assume these are similar, then basically this means that the KB must be larger than the Ka. That means binding of the incorrect ones should be much weaker than binding of the correct ones. That's what helps you make the process go on at a reasonable speed without having too much of a slowdown. Okay, let me show you this in a very nice experiment. So this is very old. This is from 1988. I noticed this is probably before most of you were born. but it's really a nice paper because they use this. I mean, problem with newer papers is we now know so much about ribosomes and about their kinetics in a lot of detail that people study them also with very, very detailed models. And we want to have a minimal description here. This Michaelis-Menten-like description, that's exactly what they do here in this paper. Let me show you what they do. They use a substrate that is in that case simply a poly-U messenger RNA. So it's a messenger RNA that has a sequence that is U, 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 and so on. And 3 U's are phenylalanine. So the protein that is, well, polypeptide that is made from this is not a real protein. It's the most boring sequence you can imagine. It's just a polymer of phenylalanine. Okay, and they translate this now with, on the one hand, with the correct tRNA for phenylalanine, but also with a tRNA for leucine, which is a non-cognate substrate which would generate the errors. And what they do here, and what is really nice, is they first translate this messenger RNA with the incorrect tRNA only. Okay, so they only take the lower branch of my 2 Michaelis-Menten schemes system. And you see, well, it looks a little bit like Michaelis-Menten, and they analyze this with one of these plots where you can map it onto some linear curve, so you can nicely read off the parameters, and you get the Michaelis constants and also the speeds, the saturation speeds of this reaction. So they use this to determine basically the VB and the KB. And the same thing for the VA and KA, that was already known at that time. There's one thing here, a note. In all these plots, something is wrong with the exponents. So there it says 10 to the 6 molar. That should be 10 to the minus 6. And the same thing, it's very systematic. In all the plots in this paper, the minus sign in exponents is missing. Anyway, so they get the parameters, and they do it for 2 different tRNA leucine tRNAs, because there's different versions. In the following, I only use this one here because, well, you see more of the nonlinear behavior, so I trust it a bit more than this one. And well, here are the parameters. So they have the Ka and the Kb. And what you see is they differ by a factor 60. So there is quite a difference. The Kb is larger than the Ka, which means the correct one binds more strongly by a factor 60. So that's already good. And you also see that in the V, in the incorporation velocity, there is also a difference by a factor 20. So indeed, the incorrect one is incorporated more slowly. Again, if you insert this into this phi, our reduction of the speed, then you see, well, if the concentrations are the same, you get a reduction of just 2%. So basically no reduction. So the system is quite nicely, parameters are very well chosen here. If you say, well, the CB in the cell should be something like 20 times Ca because you have 20 amino acids, of course, they're not exactly the same concentration. So this is a very rough estimate. But if you say, well, we have a factor 20 here in concentration, then you get a reduction by a quarter, which is still pretty good. So the reduction in speed is not as dramatic as it could be. Okay, let's now think about accuracy, because that's the other quantity that we're interested in. And accuracy, again, in our kinetic scheme here, that's very simple with these 2 parallel Michaelis-Mentens, we can say the error rate of this system, this is simply the fraction of flow that goes through the lower part. Okay? The sum of these two, this flow and this flow, that's the overall elongation. And well, a fraction of that will be incorrect, and that's this lower part here. So epsilon is just VBPB divided by VA, this should be a small v, VAPA plus VBPB. And again, this we can just write down by taking the probabilities from this type of equation over there. And what we get is this, which we can approximate by this, because, well, the error rate will be small, which means this should be large. This part here should be larger than 1, so we neglect the 1 here. Okay, so the error rate will be this VBHB divided by VAHA. And what I'm going to do now in the next few equations is I will just take this result and write it in different ways. Okay, this is the result. If we wanted to have the number, we could just calculate it. Well, we can write this in different ways, and that's somewhat instructive to do. The first way how I do this is I just reinsert what these h's were. Okay? Remember, h was a ratio of concentration to Michaelis constant. So if I insert this, what I get is a term VB divided by VA, and then I have a term that is KA divided by CA divided by KB divided by CB. And if I split it in this way, then I have one term that tells me how much my error rate comes from, or how I can keep my error rate small by the velocities, choice of the two incorporation rates. And the second term tells me how to keep the error rate small by choice of the binding. So there is discrimination between correct and incorrect in both parts of the path, in both steps. There is some part from binding and some from the incorporation. There's another way I can rewrite this. If I now, for my Michaelis constants Ka and Kb, I insert this as the definition through the rates and reorganize a little bit, then I can pull out the binding rates. And I have two factors here. One is a ratio of the binding rates in the initial step. So this is the rate with which correct or incorrect monomer first binds to the machine. And then the second step is basically a splitting probability, because after a monomer has bound, it has basically, the machine has to make a decision. Either this goes out again or it is incorporated. So you have this splitting into the two options, and the probability that it stays in is what is used here. So basically you can say, well, there is first of all discrimination in the rate at which the monomer binds in the first place, and then there is discrimination in whether after it has bound, it's incorporated or it's ejected again. So these are all just different ways of looking at the same result. I'm just taking this one equation and moving around terms, but it's useful because it gives you an impression of how this machine generates discrimination between correct and incorrect. Sorry, let's see. Wait, I'm not quite sure I got what you said. What must be small? You mean here?
Yeah, so essentially perhaps means that VBH is less than VH, much less, which is why we have the ratio, right?
Yes. So basically I'm saying, well, I'm basically coming backwards, okay? I say, in the end, this is a machine that is supposed to be accurate. If this is a machine that doesn't care about accuracy, this would be the result. But we know that it should be accurate, and that means the error rate should be small. And the only way that this thing here is small is if this term is large, And if this term is large, I can neglect the 1, and then I get the approximation. Okay, so what I do here is I kind of put in what I want to get out. I could, of course, say, well, for some reason, we now believe that VB-HB is smaller than VA-HA, but we don't really know this. What we know is this is a machine that was made by evolution in a way that is accurate. So the error rate should be small, and when it's small, then that means this part has to be large, and then we can make the approximation. Other questions? Is everything clear, or am I totally boring or losing you?
Since they are not asking question, I thought I will make one or two comments to supplement what you had said. So first was the central dogma. So Francis, you had shown Crick's paper of 1956. In fact, he wrote another one in Nature in 1970, where he clarified, because some people started saying that after reverse transcriptase was discovered, that as if central dogma has broken because information was flowing in the reverse direction. And he said that is not central dogma. Central dogma is that from protein, information cannot go backward to RNA or DNA. So that was one part you had already mentioned, but I thought, since there are students, I should mention that. The other was that you also showed that if omega 2 is small, then the ratio is just the what is called K, capital K, which was the ratio of the forward and backward. So that is actually the expression that appeared in Michaelis and Benton's original paper. It is the one that we normally use is the one that Briggs and Holden derived later. So I thought, just for sake of completeness and students, so I would make those two comments.
Thanks for mentioning this. And if you paid attention yesterday, then you have seen this yesterday. Do you remember that? At least 2 of you do. Yeah, anyway, it's not super important. It's a historic thing. But yeah. Okay, let's get back here. So I have written this in 2 different ways, and now I'm writing it in a 3rd way. And the 3rd way is making use of exactly what Debashish just mentioned, namely this approximation that the omega 2, which now is called V2, VA and VB, the rate of the irreversible step, that this is relatively small. And if this is small, then I can take these Ka and Kb and replace them by the dissociation constants. Okay, and the nice thing is that now the dissociation constants, these are equilibrium properties. Yeah, this has to do with binding of 2 molecules, and we can relate this to an energy. Somehow I'm missing a plot here. Whoop. Okay, this is totally messed up. Okay, this I still need. Okay, so I'll replace the Michaelis constants with the dissociation constant by this assumption that these incorporation rates are small. And well, then these dissociation constants, if you imagine this is just unbinding of 2 molecules, then you can imagine this is some transition. No, I need that later. Sorry. This is this term here. If this is just binding, then basically the difference, this ratio of the 2 dissociation constants is a Boltzmann factor of the difference in binding energies. Okay, the dissociation constants are related to the binding energy, Boltzmann factor with the binding energy, and if I have a ratio of these 2, this will just give me a Boltzmann factor of the difference in binding energies between correct and incorrect monomers. So now I have an expression where I can relate this part here, the discrimination by binding, to an energy. So this is the energy difference between binding a correct amino acid and binding an incorrect amino acid in the case of the ribosome, or actually it's the tRNA in that case that is matching correctly or not correctly. And a correct or incorrect nucleotide in the polymerases. Which also tells you that the difference in this discrimination here cannot be huge, because the different nucleotides, they are similar molecules. Okay, so if you match a wrong one, then maybe you miss 2 hydrogen bonds or something like that. It's not a gigantic difference in energy. Okay, the other part here, we still have this part here about the VB and VA, which is the incorporation rate. This is a reaction, irreversible reaction, and this we can imagine with this nice energy landscape. So assume this is before the reaction happens, there is a barrier, and then after the barrier, the ribosome has stepped forward, and the new amino acid, the chemistry has happened, is incorporated into the chain. Okay, and this type of chemical reaction, you can just imagine, well, the VA or VB, this is basically some constant times an exponential, this Arrhenius type factor where you have the barrier height. Okay, that's the forward rate. And if I now take the ratio VB divided by VA, then I have this, well, going over one barrier and going over another barrier. And basically I can write this as an exponential of the difference in barriers between the correct and incorrect monomers that are inserted. So imagine you have a binding that is matching versus a binding that's not quite matching, and then the chemistry happens and everything has to move through these small channels in the molecules. So you can imagine that maybe the barrier for the incorrect one that's not fitting quite exactly is higher than for the one that's fitting exactly. Again, this tells you again, there is one part here that is related to equilibrium properties, binding energies, at least in this approximation. And there's the other part here that has to do with a kinetic property, with the barrier in this reaction of incorporation. Okay, we still have the concentrations, but what I will do is I will again assume that these concentrations are very similar, and this is approximately 1. Of course, in a test tube, we could make the concentrations different, but in the cell, you can assume these are similar. Or actually, typically, there will be more incorrect ones than correct ones, because there are different options of how to make a mistake. Okay? There are 19 wrong amino acids compared to 1 correct one. There are 3 wrong nucleotides compared to 1 correct one. But let's not worry about this and just say this is roughly 1. So we have this difference here in barriers, and we have this difference in binding energies. Okay, let's go back to the experiment, because in principle we have everything in this experiment. We have everything, all the parameters. You see up here the decrease in speed. So this is the speed of the reaction plotted as a function of the incorrect monomers, and the concentration of the incorrect ones is very high here. I think it's the same. It's 10 times higher than the correct ones, somewhere here near 1. Okay, so this is a very high concentration, and we said before, well, with 20 times the concentration, incorrect ones has 20 times the concentration of correct ones, we expect a decrease of 25%. It's close to that. And below you see the error ratio. And well, first of all, the error ratio increases linearly with the concentration of incorrect ones. That's what it should be if you look back here.
Stefan?
Yes.
In the previous slide, what is the x-axis?
This one?
In the top panel, what is the x-axis?
It's the same x-axis. This and this has the same x-axis, and it's the concentration of the incorrect monomers.
And then what are these two circles, empty and
Oh, these, okay, there are 2 different types of incorrect ones.
2 types?
Yeah, if you go back, well, let's go back to the plot before. This one. There was this tRNA leucine 4 and leucine 2. These are both not the correct ones, but they're different. They incorporate the same amino acids, but they're different tRNAs. And you see this one incorporates better than this one. And so they have done basically the same experiment with 2 variants of incorrect ones. But I have only shown you the parameters for the filled ones. No, for the open ones, sorry. Okay, so below you see the error rate, basically the fraction of incorporated leucines, and let's estimate what it should be. We said the VB over VA, that was a factor 20, if you remember, and the Ks, that was a difference, that was a factor 60, and I just told you the concentration is, the B has a concentration that is about 10 times larger than A. Now I need to do this in my head, so 60 times, what did we do? 60 times 20 divided by 10, yeah. Should be something like 10 to the 3 or so. Anyway, this is what it should be. This is what we actually, at the point that they would get at the concentration that we just estimated. And I think if I remember correctly, if we estimate this, we get something like 100, which is up here. So it's not exact, but I mean, it's off by a factor 3, which is not so bad. I mean, the order of magnitude is right. And what this probably tells you is that, I mean, we know that the ribosome is much more complicated than a simple Michaelis-Menten kinetics. There are many steps and there are many more details in this kinetics, but at least it gives you a rough estimate of the order of magnitude where the accuracy is here. Okay, and it also tells you, well, we have a factor, we said a factor 20 from here and a factor 60 from there. So the 2 contributions to the accuracy, they're of a similar order of magnitude here. Okay, it's not that one of them is dominant and the other one can be ignored. Which also makes sense because, I mean, when you have these 2 options, why not use them? Okay, how are we doing in time? Pretty good, I think. Yeah, very good. Okay, maybe at some point you have heard about proofreading. And so what I need to tell you now that everything I've told you so far ignored proofreading. There is no proofreading, okay? I just talked about incorporating by binding and chemistry happening. Okay? And the idea of proofreading by kinetics, that's maybe one of the most brilliant ideas that theorists have contributed to biology. Because it shows up again and again everywhere. And I think at the time when this idea was first proposed, basically simultaneously by Hopfield and by Nino in 74, 75, that was a theoretical idea. But this has then later been found in many, many systems. Okay, so what I want to do in the rest of the time is give you a little bit of an overview of proofreading. And the basic idea is that you spend energy to improve the accuracy. Okay, you put more energy in and use that to improve the accuracy. What I will show you here is the scenario that Hopfield proposed. Nino had a slightly different one, but it's actually closely related. So this is our scheme that we had, except that I put some space here. We have this reversible binding plus irreversible chemistry, and we have a correct path and a lower path that is the incorrect one. And what Hopfield said is, well, we make this very simple. We say the only discrimination is in this unbinding rate. So these V's, my VA and VB, these are the same. Also my binding rates, the omega A and omega B, these are also the same. The only thing that is different is this rate here. So the only discrimination is in unbinding. And he also makes the assumption that these reactions are much faster than this one. Okay, so the K's can be replaced by the dissociation constants. And then my error rate can be written, now this is already the result. Well, I'm missing an equation here, so let me write this equation on the board. So if I do this, if all the rates are equal, let's go back here. This is our equation. Okay, this is what we have. If now in addition VA and VB are the same, the only thing we're left with is this Boltzmann factor. Okay, so without proofreading, our error rate is simply, well, let's write approximately e to the minus this delta of the binding energies over kT. This is what Hopfield uses as his reference case. And then, I think the order in which the things show up on this slide is not quite correct. Anyway, the idea is, let's introduce an additional state here. So after the monomer has bound, the substrate has bound, we drive the system into some high energy configuration. So here we put in energy to drive the system into this configuration A star or B star, and only from there the chemistry happens. Okay, so far we haven't gained very much, but we gain much if now the substrate can still unbind from this state here. Okay, so we have substrate binding. Afterwards, some configurational change that puts the system into a higher energy configuration, and from there the chemistry happens, but from this higher energy configuration, the substrate can still unbind, both the correct one and the incorrect one. Why is it important that this is higher energy? What do you think? Any guess? Why do I put in energy to get here? No, it's not really a transition state, but it's somewhat similar in the sense that you have a state that is between this and this, but it's not really on the path of a reaction, but it's rather some intermediate. Which arrow is missing here? Could you draw an arrow in this plot that I haven't drawn? No. A star to A, okay, yes, this we could have. Yeah, and we're saying, well, this we don't have because that's where we put in energy. Well, at least the rate is very small, so we drive this strongly towards A star. Free to A star, exactly. This one is also missing. Okay? And what would this be? I mean, this would be binding of a substrate directly into this intermediate state. And that would be bad because then we don't have this, we don't have this state here where we have the discrimination between right and wrong. Okay, so we drive this into a high energy state so that binding from free to here doesn't happen. That's very strongly suppressed because that would need energy. You would need a huge thermal fluctuations to bring the system there. Okay, so we put in energy to drive the system in here, and since this is now higher energy, the system cannot get in there without going through A first. That's important. And then if you take this now, okay, now we have 5 states, which means we have a system of 5 equations. We can calculate this again. It doesn't look very nice. You can do this in all generality, but then if you use all of Hopfield's assumptions, most of the things drop out, and eventually what you get is 2 terms. One is this term here, the ratio of the dissociation rates. This is what is unbinding, and then we have a ratio of these rates here. The omega, okay, omega minus A was this one here, and the K minus A and minus B are the new ones. Okay, and both are unbinding, and the idea is unbinding differs in this binding energy, so now we get 2 factors of this Boltzmann factor. And if the Boltzmann factor is what makes the error rate small, now we have it twice, so the error rate is even smaller. Okay, that's how this works. You put in energy here, you get a second factor, and then, well, you have this increased accuracy. Of course, this scenario is very much simplified because we have, for example, assumed that these 2 rates are the same, and we have seen that they are different. So actually, having these rates different in addition, that will give additional discrimination. Basically, now when the system comes in, it has a chance to unbind from here. That would give a discrimination between right and wrong. And then it has a second chance where the incorrect one unbinds preferentially when it goes into this state. And then maybe there is additional discrimination from the different incorporation rates. Okay, so that's a key idea, I think, in theory applied to biology, that you can put in energy into a system and then you get additional accuracy in a reaction. And if this is very useful because this binding energy, I said, well, these are molecules. These are similar molecules. So first of all, the binding energies are pretty much given. These are just constants. And if you have 2 similar energies, their binding energies will differ, but they will not differ dramatically. We will have a difference of a few kT. So e to a few, that's maybe a factor 10, maybe a factor 50 or something like that. But if you square this, then that increases quite a bit. So that's very useful. And if you get an additional factor from here that's of the same order of magnitude, that's also useful. Okay, let me briefly talk about a few more variants. So this was basically Hopfield's more or less hypothetical scenario at the time. And at least in the ribosomes, the scenario that you have is somewhat similar to this, that you really have binding of the tRNAs, you have an initial discrimination by binding, and then you have a separate proofreading step happening afterwards that is driven by GTP that comes in with the translation factors. So in that case, the proofreading state is something that is on the forward pathway towards the incorporation. It's something that you have to go through in every step. But in other enzymes, and RNA polymerases and DNA polymerases are of that type, you don't have the proofreading step really on the same pathway, but rather this is something that branches off of the main pathway. And well, since so far all of this is basically things that's kind of textbook, I thought on the last slides I'll at least briefly mention a few things where we have worked on ourselves. And one of them is RNA polymerase and the other is DNA polymerase. And both these things were done together with Mamata Sahu, who's at the University of Kerala now, but she was when we started this, she was in my group, who at the time was still in Potsdam, because that happened long ago. Anyway, and one system that we looked at is RNA polymerase. And RNA polymerase is a nice example where you have error correction that branches off the main pathway. Okay, so you have a scheme in which basically now this is our path here, like this. And you have a step that goes away perpendicularly, so to say, and then maybe you go back here. And of course, the same thing down there. Okay, so what does RNA polymerase do for error correction? It's actually even more complicated than just this. So RNA polymerase steps forward on the DNA, and if it makes an error, what happens is that it starts moving backwards on the DNA and simultaneously on the RNA. So if you imagine the RNA polymerase has the DNA along which it moves, and then it has made an RNA that is copied up to that point. And then it slides backwards on both of them. Okay, which is different from what it normally does. Normally it moves forward on the DNA and then it extends the RNA. Now it doesn't reverse that, it doesn't go backward on the DNA and shortening the RNA, but rather it moves backwards on both of them. So if it moves backwards on both of them, then this motion actually is also not directed, but it's diffusive, so it basically makes something like this. And eventually what it will do is it will just cut off all the RNA that sticks out in the front. Okay, this is called backtracking and transcript cleavage. This is what you see here. In our drawing, you have the RNA, so it moves backwards on both of them. The RNA sticks out here in front, and then it will just cut off everything that sticks out in front. And the error, if it starts moving backwards shortly after an error, then the error will be in the part that sticks out and is cut off. Okay, if I simplify this was the drawing that we made at the time, and it doesn't look quite as nice like this, so I try to redraw it. This is this is basically the same as this. Okay, we have two Michaelis-Menten schemes, the correct one and the incorrect one, and now the A star state, so to say, is not between A and 3, but rather it branches off to the side, and then you can go back here. Also, the A star is not really one state, but it's this diffusive backward motion, and likewise for the B star. But basically, you drive it to the side here, and then you have a way of cutting off and going back to the start. The start typically is 2 bases back, so it doesn't just cleave off 1, but typically it cleaves off 2. That's what the molecule does. And what happens in this case, you can still do the same type of analysis. You now don't get a product like in Hopfield's example where you have 2 factors that discriminate, but rather you get a sum here. So it's a bit more complicated than Hopfield's scheme, but it still improves the error rate. And the other thing I wanted to show you is DNA polymerase. And there again, you have something that looks very similar to this RNA polymerase case. Actually, this model that we have analyzed, and this is something we did rather recently, the model that we have analyzed here is actually a model that goes back to work by Ajit Sharma and Debashish Chowdhury a few years, well, also 10 years ago. So in that case, in the DNA polymerase, you also have a branch to the side. And the branching to the side now is not a backward motion on the copy, but rather it's a transfer of sort of the new end of the new DNA from one side in the enzyme, which is the site for polymerization, to a site that's called the exonuclease site. Sorry, there's a typo here. It's exonuclease site. This is the site where this new nucleotide at the end can be cut off. Okay, but other than that, it's basically the same. You don't move backwards, but you transfer the polymer from one site in the enzyme to another site, and then you can cut the last nucleotide off. And well, what we saw when we analyzed it here is that we really need this discrimination in the stepping rate, in this forward reaction rate. Here it's called epsilon. That's what was the V in the model before. Okay, this rate in the chemistry, in the incorporation, we really need the difference for discrimination for two reasons. On the one hand, this is what allows the unbinding to happen. So if the incorrect one is incorporated more slowly, then it has more time to unbind again, while the correct one, when it's bound, is basically immediately incorporated. But not only that, we now have this proofreading pathway which branches off, and that also needs time to happen. So if the incorrect one now, the chemistry is slow, that means there is time for this transfer to the other side where the error can be corrected. While in the correct one, the chemistry is fast, that means there is no time for the transfer because you don't want the transfer. You don't want to correct the correct ones. You don't want to cut them out. Of course, that will happen stochastically from time to time, but it should happen, well, as rarely as possible. Okay, so we need a difference in this forward rate because that gives time for both processes that really discriminate. But it's actually not enough, and to really get a high rate of accuracy here, we also need that this cleavage rate, the rate at which the last nucleotide is cut off, is different for the correct and incorrect ones. So this also needs to discriminate. Anyway, I don't want to go into the details here. What I wanted to show you with these examples is that in different enzymes, the mechanisms and the details of the chemical kinetics can be very different, but they all have these additional steps that give additional discrimination by making by having more reaction steps where correct and incorrect monomers have different rates. Okay, I think that's where I wanted to stop for today. Well, we still have 15 minutes, that's good. So let me point out a few key points of what I've talked about. First of all, I think what we have seen is that the accuracy of these polymerization reactions is achieved by a combination of different contributions. There is a contribution from binding initially. Correct ones bind more strongly than incorrect ones. But there is also a contribution from the reaction rates themselves, so from the incorporation. Okay, think of this again as a difference in binding energies and a difference in energy barriers in going forward. You have both, typically. At some point I mentioned that there is usually this trade-off between speed and accuracy. So increasing the speed very often goes at the cost of decreasing accuracy. And then a very important point at the end, that kinetic proofreading is a very general scheme, but that can be implemented in different versions, in different enzymes, in which you increase the accuracy of a process by putting in additional energy. Okay, I think at this point I'm done. Maybe I'll very briefly show you 2 slides because that's what we have a very nice PhD program in Germany now, which is new. So if you know any undergraduates, so for you this is too late, but if you know any undergraduates who are very good and interested in going to one of the top US universities or so, maybe you tell them there is a good alternative. They can also go to Germany. There is this thing called the Max Planck School Matter to Life. And it's an interdisciplinary program for masters and PhD It involves some biological physics, some chemistry, and synthetic biology. And it's done not in one university, but in a network of many places, many universities. And one of the key places is Göttingen. The other key place is Heidelberg. And it involves, for example, also a lot of Max Planck institutes and other research institutes all over Germany. Everyone who's interested in understanding the building blocks of life and understanding and engineering lifelike systems. So if you know someone who's interested in this, let them know. They can contact me or have a look at our webpage, which I show you here. Okay, enough of the advertisement. But if you know someone, tell them. I'll put up my key points again, and I'll stop here for today. But I hope you still have questions, because we still have time, and so we can discuss a little bit. So thank you very much for now.
Thank you, Stefan, for this very clear and thorough talk. So we have time for a lot of questions, I guess.
Do I moderate or do you?
Yeah, I have.
Go on.
So I want to ask this question. Because you talked about this regulation. So these reactions, do they have this auto inhibition? Like if there is some concentration, then there is a cutoff, then this reaction will not happen, or like that.
Which reaction do you mean? So in like, transcription and translation?
Yeah, means translation, because it is protein concentration. Like if
I mean, there can be auto inhibition through transcription factors. So if the protein that is made through the process is a transcription factor, then it might downregulate its own transcription, for example. But yeah, that usually works at the level, or in most cases it works at the level of how the process of transcription is started. And what I've talked about most of the talk is really the process, how it proceeds after it has started. So yes, it's there, but it works at the beginning of the process. In most cases, there are exceptions, but the typical cases is in the beginning. You also had a question.
Ribosomes, RNA polymerase, and DNA polymerase, all of them have two-feeding mechanisms, right? But still, the error that they make, right, it is very huge difference, right? Ribosomes are very error-prone. They make error 1 in 10 to the power 4 incorporation. But DNA polymerases are very accurate. So what is the difference between them? Why, DNA polymerase is more accurate than ribosome?
Okay, that's a good question. First of all, the statement that the error rates are different between the enzymes, that's of course correct, and that I think you can explain it functionally, because errors that you make in DNA replication are kind of permanent. These are errors that will stay. I mean, these are errors that you pass on to your children, and well, if they are bad, then the offspring will not live, and if otherwise you will maybe generate mutations. While if there is an error in a protein, then well, one protein is wrong. So I think on a functional level, it's clear that high accuracy is even more important in DNA replication than it is in transcription and translation. How the difference is realized at the molecular level, that really depends on the design of the enzymes, and I can't really tell you why that is, but yeah. I mean, I think it makes sense that evolution selected for very correct DNA polymerases, and maybe the pressure was somewhat less on the ribosome, for example. I can also imagine that on the ribosome, if you think of a speed-accuracy trade-off, there is more pressure on speed, because this is something I will probably talk about the day after tomorrow. Ribosomes are kind of expensive machines that you want to work as efficiently as possible. So maybe compromising a little bit on accuracy there allows you to go faster. So you would probably drive more towards speed and less towards accuracy. Somewhat less. Not too much, of course. So I think at that level, I find that relatively understandable. At the molecular level, I'm sure there are very detailed studies that one could look up, but I don't know them at the moment.
Yeah, Stefan, a related question. See, after all, ribosome has the RNA components, ribosomal RNA, and evolution actually might have brought it from there, that later proteins were added and so on. So it is possible that in the prokaryotes from where it might have come, or ancient some other ancestors, so there the accuracy high was not needed so much. So is it possible that, since actually ribosome has that strong RNA component, so the error level remained quite high and it was tolerable, whereas the proteins which became the DNA and RNA polymerase, they came probably later and the accuracy was needed. So is it possible that ancestry wise also there is something other than just functional requirement at the level of eukaryotes at a later stage?
I mean, the honest answer is I don't know. I think it's possible, but it's also a very strong statement if this is correct. So
I don't know. I'm just wondering, because the other two are just proteins, but
yes.
ribosome has ribosomal RNA.
Yes.
And the ancestors, of course, at some point was RNA world, so from that it might have come.
I think it's possible, but then the accuracy of the DNA polymerase is also higher than of the RNA polymerase, and they are both proteins. So maybe there is a contribution, but I really don't know. But it's an interesting speculation. It's something one should think about.
Any other questions? If that is not the case, let us thank Stefan.